Good evening, one and all. Today we are going to see about an important general topic with respect to our subject. It's the various methods of propagation of crude drugs, various methods of cultivating the crude drugs. So, in this topic, we are going to see by which methods or what and all methods we can use to cultivate medicinal plants in our field, right? So the various methods that can be used to propagate medicinal plants are of three types. One is called as sexual propagation, number 2 is called as asexual propagation, number 3 is called as aseptic propagation. So number 1 is called as sexual propagation, number 2 is called as asexual propagation, and number 3 aseptic method of propagation, right? So we'll see one by one. Sexual propagation, it's also called as seed propagation. We can call sexual propagation also as seed propagation. Why means, as we all know, seed is the sexual organ of the plant. So if you are using seed and propagating a new plant, it's called as sexual propagation and will be also called as seed propagation. So if a new plant is coming out of the soil with the help of seed, due to seed, then that particular plant will be called as a seedling, right? So if you are raising a new plant with the help of seed, the plant will be called as seedling, right? So we'll see some general criteria about the sexual propagation. The seed what we are going to use should be of good quality, yes, right? Which is for two reasons. So if we are using good quality seeds, then the germination of the seeds will be very good. And if we are using good quality seeds, the plants will be having proper uniform growth and hence has good amount of phytoconstituents, right? So whatever the seeds we are using should be of good quality. The seeds should be free from insects, diseases, used seeds and other seeds. So what's the seed we are going to use for germination? What's the seed we are going to use for propagation? It should be free from insects or other foreign matters. It should be free from any disease. It should not be already used seed, and other plant seeds should not be present. So if the seeds are free from disease, then ultimately the plant will have proper growth. If the seeds are free from insects and other foreign matters, ultimately the quality of the seeds will be good. If you are using already used seeds, it won't germinate, so we won't be having good yield. And if other seeds are present, then ultimately the crop will get adulterated. So whatever the seeds we are using should be free from insects, disease, and it should not contain used seeds. Okay. Long storage of seeds should be generally avoided. So if you buy seeds from the market, it has to be used within quick period of time and it should not be stored for longer period. Why means, if you are storing it for longer period, it may go to dormant stage and ultimately the germination capacity will get reduced. So long storage of seeds should be generally avoided. If the seeds are not used in the near future, it has to be stored in the cool and dry place. So if suppose if we think that we are not going to use the seeds for a certain period of time, then we have to dry it properly. It has to be cleaned. Then we have to pack it tightly and should be stored in a dry place so that it will be having proper germination capacity when it is used, right? So the germination rate of the seeds can be tested by rolled towel test or excised embryo test. So if you want to check the germination capacity of the seeds what you are having, You can check it by doing rolled towel test or by doing egg-sized embryo test. Okay. So in rolled towel test, we will take a cotton cloth. We will keep certain seed sample which will be closed and kept in a dark place. After certain period of time, we will open and see how many seeds has germinated. So if many seeds germinated means, then ultimately the germination capacity of the seed what we are having is high. If very less seeds has germinated means the germination capacity of the seed what we are having is less, right? So by doing these tests we could able to understand whether the seeds has good germination capacity or not, right? So now we will see how to enhance germination capacity. If suppose if the seeds are not germinating properly or if the seeds are not having good germination capacity, then the germination ability can be increased by doing certain treatments. Okay, so we will see certain examples. By soaking it in water for a day. So by soaking or by keeping the seeds in water for 24 hours, the germination capacity may be enhanced. So the best example is for castor seeds, we can do this type of treatment, right? By soaking it in sulfuric acid. So as like soaking it in water, we can soak the seeds in sulfuric acid for certain period of time, by which we can increase the germination capacity. And this is generally done for henbane seeds. And same like that by spraying gibberellin. So if suppose you had stored the seeds for longer period of time and you think that the seeds are in dormancy, then you can spray the gibberellin, which is a plant growth regulator, right? It is a plant growth hormone. So if you spray gibberellin, then ultimately this gibberellin will break the seeds dormancy and hence the germination capacity of the seeds increases. So by doing any of this treatment, the germination capacity of the seeds can be enhanced, right? Good. So next we'll see what and all advantages we are having if we are doing seed propagation. So seed propagation is very cheaper. So when compared to asexual propagation, we will be having less expense in sexual propagation, so that it's cheaper. It's easy to raise. When compared to asexual propagation, in sexual propagation we can put a seed and we can able to raise a new plant very easily, right? The plants are long lived. These plants are generally lives perennially when compared to asexual propagator plants, right? The life period of seed propagator plants are much more when compared to asexual propagation, and bears more heavily, which means the fruiting capacity of these seedlings are very high when compared to asexually propagated plants. Good. What are the disadvantages? The growth will not be uniform. So based on the germination capacity and other factors of a particular seed, the growth of the plant may be very quick or very slow. So each and every plant what we are propagating with the help of seed will not have uniform growth. Okay, that's one disadvantage. Number 2, bears fruits after longer period of time. It bears heavily but after a longer period of time. And harvesting cost is high. So raising a new plant is easy, but harvesting and remaining needs high expense. So these three are the disadvantages of sexual propagation, right? So next, asexual propagation. This asexual propagation is also called as vegetative propagation. Why means here instead of using seeds, we will be using any part of the plant and propagate a new plant. So in asexual propagation, we will be using a plant part to produce a new plant. For example, as like in hibiscus, we will be using a stem cutting. to produce a new plant, right? Like that we may be using root cutting, we may use tubers, or we may use even leaves. Like that, if you are using a plant part and if you produce a new plant, then that will be called as asexual propagation. So here are some examples I had noted. Example from bulb are using corn, tuber, rhizome, runner, offset, sucker, stolen. So like that, many things are there, right? So if you use any of these plant parts and propagate a new plant, then that will be called as asexual propagation. Since here we will be using plant parts, it can be also called as vegetative propagation. Right? What's the advantage? Here the growth will be uniform. So in sexual propagation, growth will not be uniform, but here the growth of the plant will be uniform and it bears quickly. So in sexual propagation, it bears slowly after a longer period of time, but in asexual propagation, it bears very quickly. And the disadvantage, its living period is less. Right? In sexual propagation, the plants are long lived, whereas here the plants are living for a lesser period of time. Right? The last method, aseptic method of propagation. So if you are using a plant cell or tissue, if you are using a plant cell or a tissue and produces a new plant, then it will be called as aseptic method of propagation. So in this method, we will be producing a new plant in a lab, right? In a lab setup, in aseptic condition, we will use a single cell or a group of cell and produces a new plant. So in simple, we can say this technique as plant tissue culture. Right? So by using tissue culture technique, we will be propagating new plants, and when these new plants attain certain height, it will be transplanted in the soil. So this plant tissue culture, we are having a separate chapter in which we will discuss about it elaborately. Right? So that's about today's topic. Thank you.